So when we're looking at this particular replication fork, which is moving this direction, this strand is being built. This is the primer, and then we're adding nucleotides, building in this direction. So that's the leading strand. It's built as a continuous sequence. The lagging strand, as this comes apart, it has to unwind, and it'll get to a point where another primer can be bound, and then the building has to happen in this direction. So from here, that was built, then it unwound some more. From here, that was built, then it unwound some more. From here, that was built. But we have a problem on the lagging strand because the lagging strand now has RNA in it. This red color is indicating that that's RNA, not DNA. We're supposed to be replicating DNA, so the cell has to get rid of those little RNA primers that are on the lagging strand. It also has to get rid of the one at the origin. So every single Okazaki fragment begins with a short RNA primer. It's got to be removed. This is going to be accomplished by DNA polymerase 1. So remember that DNA polymerase is a complex molecule. DNA polymerase 3 is the primary builder. DNA polymerase 1 is going to come and take those RNA nucleotides out. But it still leaves a gap. Once the RNA is removed, you still have a gap between what is DNA of one Okazaki fragment and DNA of the next. And although DNA polymerase 3 can add to the end, it cannot connect those two pieces together. So we have to have another enzyme. An enzyme that can form a phosphodiester bond between sections of DNA is called ligase. And I kind of like the name of that enzyme because when you think about ligation, if you've ever been through a surgical procedure where they do ligation, they're tying things together. Or think about ligaments. Ligaments tie bones together. Ligase, to me, sounds sticky. It's glue. It's the enzyme that catalyzes the phosphodiester bonds, connecting fragments of DNA together. So whenever you see the enzyme ligase, again, ASE ending tells you it's an enzyme, and think of connecting things, like a ligament, or like ligation. Anyway, so now, this diagrammatic representation shows you some of the enzymes that are involved in DNA replication. There is a problem with this diagram, and I'm not sure why it was put together this way, but instead of having DNA on the DNA polymerase 3, it's ADN. It's like, seriously? It says DNA here, where it talks about DNA, parental DNA, replicated DNA. Like, why are we calling it ADN? I don't know. It's DNA polymerase. DNA polymerase 3 is the primary builder. So what we see in this diagram, primase builds the primer. At the origin of replication, there's one primer on the leading strand to start that building process. On the lagging strand, there's going to be multiple primers, and primase is always going to be building those. Then you have DNA polymerase 3 that can add nucleotides to an existing or to a free 3' end. You have to have DNA polymerase 1 to take out the RNA, and then you have to have ligase to connect the Okazaki fragments together. That's not mentioning helicase and topoisomerase that aren't shown up here. So there's a bunch of enzymes. So again, DNA polymerase is multi-part. 3 is the primary builder, 1 takes out the RNA, 2 does some other repair stuff. So again, just a review, DNA replication requires 1, DNA as a template. You have to have that template strand to determine the nucleotide sequence. You have to have enzymes. The enzymes you need to know are primase, which is actually an RNA polymerase, but primase builds the primer. Then you have to have DNA polymerase. DNA polymerase 3 is the primary builder. DNA polymerase 1 takes out the RNA pieces of the Okazaki fragments. You have to have ligase to connect together the Okazaki fragments. Those are the primary enzymes that you need to know. Helicase just breaks the hydrogen bonds, allowing the strands to separate, and topoisomerase, which is also called helicase. What did I say? Oh, gyrase, I'm sorry. So topoisomerase is gyrase. It just prevents the DNA from supercoiling ahead of where it's untwisting. then energy. That's the other requirement, because this is a synthesis type reaction we're building. The energy is supplied by nucleoside triphosphate. If you're building DNA, you have to have both dNTPs and rNTPs. The rNTPs are necessary to build the primers. So again, not just ATP, but ATP, GTP, CTP, etc. All the different bases have to be associated with nucleoside triphosphates. So pay attention to the lowercase letter in front of NTP. NTP stands for nucleoside triphosphate. The D or R in front of it tells you what the sugar is. If it's a dNTP, the sugar is deoxyribose. If it's an rNTP, the sugar is ribose. And the bases are going to be different. Again, these are high energy compounds. They have pyrophosphate. The pyrophosphate, when that bond breaks, when you pull off two phosphates to put in one nucleotide, that releases a lot of energy, and that energy is used to build the phosphodiester bond. Okay, now we're moving on to the next topic. So that was kind of summing up or finishing up DNA replication. This word, transcription, if this were not a biology class, what does transcription mean? Anybody? It's writing things down or copying information. There used to be people that were hired as medical transcriptionists, and their job was to decipher what physicians had written and rewrite it so that somebody could actually read it. They had to be really good at deciphering what the physicians were putting down. At this point, most of that recording is being done with computers, and so it's not as critical that there be somebody to decipher somebody's handwriting. But transcription basically means rewriting information. In a biological sense, it means the same basic thing. Transcribing is rewriting information, but specifically, we're rewriting information that's stored in DNA. We're rewriting it as RNA. So transcription is RNA synthesis. Information carried in the DNA is going to be copied or rewritten. It's important that you pay attention to words that you might already know. You should have some understanding of what transcription means. And then don't forget that when you see this word show up on a quiz. What is transcription? It's RNA synthesis. It's copying information that is stored in DNA, copying it as RNA. So it's RNA synthesis. Just rewriting or copying written information. So transcription is RNA synthesis. We're building RNA molecules. It's going to be like replication in that it still requires DNA as a template. It requires enzymes, and it requires energy. And it turns out the processes are very similar, except we're going to end up with a different product. So transcription is going to happen wherever DNA is. It's in the nucleus of a eukaryotic cell, it's in the nucleoid of a prokaryotic cell, but it's also associated with plasmids. Plasmids are small extrachromosomal loops, so they're not part of the chromosome. It occurs inside mitochondria, it occurs inside chloroplasts. Wherever there's DNA and information that needs to be copied, that's where transcription can take place. So a lot of different places. And again, transcription requires the same kinds of things that replication requires. It requires DNA as a template or pattern. The nucleotide sequence in DNA is going to determine the nucleotide sequence of RNA. It requires enzymes to catalyze reactions. The same kind of enzymes that were required to take the strands of the DNA apart and prevent the DNA from getting tangled up. So you have to still have the helicases and the gyrases or topoisomerases to get that DNA apart. But you don't have to have DNA polymerase, because we're not building DNA. You don't have to have primase, although you do have to have RNA polymerase. And you don't have to have ligase, because when transcription occurs, only one strand of the DNA is copied. Don't copy both strands at the same time. And of course, you have to have energy, and that's supplied by rNTPs. If you're building RNA, you don't have dNTPs in the process. So replication requires both dNTPs and rNTPs, but transcription, just rNTPs. Again, what does the little r stand for? Ribose, the sugar of RNA. So they look the same because they're just stars with letters on them. And again, only one strand is copied. However, typically, depending on what kind of cell you're looking at, or it gets even more complicated when you start looking at viruses, what portion of the DNA is being copied at any one point in time can be quite variable. So you can't say that one strand is always copied and the other strand isn't. There's information on both strands, and it's not the same information because the strands are complementary. They're not identical. So you actually have double the information content because the DNA is a double-stranded molecule. So only one strand at a time is copied, but it can be either one. This is showing a representation of transcription. The DNA strands are separated. You can see the nucleotide sequence of the DNA. And then you see this strand up here is being copied. This is called the sense strand. It's not the strand where transcription is actually happening. But if you look at the nucleotide sequence of the RNA transcript, you'll see that the nucleotide sequence matches the sense strand. It's complementary to what's called the antisense strand. And again, it's not always the same strand. It can be either one, depending on what's going on. So the primary enzyme that we're interested in when we look at transcription, again, there's helicases and topoisomerases involved, but we're looking at the primary enzyme that's building RNA, and this is called RNA polymerase. Like DNA polymerase, it's a complex. We're mostly interested in prokaryotic cells, so we talk about RNA polymerase. I tend to concentrate on prokaryotic RNA polymerase, which is a multi-part enzyme. So these enzymes are globular and definitely quaternary. They have multiple parts to them. So here you can see a whole bunch of subunits. There's two alphas. They were initially called alpha, now they're alpha 1, alpha 2. Then there's a beta and a beta prime. There's a little bitty omega, that little gray thing in the back, and the pink one that's in the front is called sigma factor. We're mostly interested in sigma factor because sigma factor is an initiation factor. Without sigma, transcription can't start. So it's going to determine where transcription is going to begin, make sure that the right strand is being copied, and that the copying process occurs in the right direction. So we need to pay attention to that pink sigma factor there. The rest of the enzyme, the alpha, alpha, beta, beta prime, omega, is sometimes referred to as the core enzyme. So you can kind of think about this as being a work crew, where sigma factor is the kind of like the foreman. Sigma factor is going to bind to a specific region on DNA that's called a promoter site. There are multiple different sigma factors, and they bind to different promoters. They tend to have an affinity for different promoters. And in these diagrams, you'll often see a promoter site is represented by a very short segment of DNA. Really, the promoter is long. It's between 100 and 1,000 bases. So it's a big, long section of DNA that sigma factor is going to recognize and bind to. If you get into the greater details of DNA, so the structure of DNA and the process of transcription, you may see reference to what's called a TATA box. So TATA box is that T-A-T-A-A-T, and then there's sometimes there's parts that are referred to as cat boxes and so forth, and never mind. That's not something you guys have to know. I do want you to know that RNA polymerase has a core enzyme, multiple parts, and it has sigma factor, which is like the work foreman deciding where the job's going to start. Make sure that it gets started properly. Now, sigma factor doesn't stay with the core enzyme. Once transcription is initiated, sigma factor kind of leaves. Again, that's why I like the foreman analogy. Oftentimes, if you have a work crew, the foreman kind of comes and says, here, this is where you're going to work, and this is what you're going to do. And then, oh, by the way, I've got a phone call. I have to go deal with another work crew over here to get them started. The sigma factors just get the core enzyme bound to the DNA properly and get the process started. And again, there's different sigma factors within cells. They have different affinities for different promoter sites. There's a lot of detail to this that you guys don't need to worry about. Just recognize that sigma factor binds to a promoter. That's where transcription is going to start. So you may hear also reference to housekeeping genes. Housekeeping genes are genes that encode constitutive enzymes. The enzymes the cell has to have, they're always present, the cell's always making them. And so typically there will be a primary sigma factor that's in charge of those housekeeping genes and making sure that those are always going to be transcribed to make RNA, and then the RNA is going to be translated. So there's a bunch of other factors also. There's a lot of regulation relative to transcription. Again, you don't have to worry about all the details. We're just looking at RNA polymerase, the core enzyme, and sigma factor. And the other thing about transcription is you have to recognize it's going on pretty fast. We said DNA in E. coli, the E. coli chromosome can be replicated. It takes 60 minutes to replicate the whole thing, but you can start a new replication cycle every 20 minutes. In between replication, transcription has to happen. And it's not just one piece of DNA that has to be transcribed, it's many pieces of DNA, because the cell has to be constantly making RNA to make the proteins that it needs to keep functioning. So all of this business is going on pretty fast. So this is going to show you a movie of transcription, and it's kind of interesting because it's all in animation. It's not even in real good focus, but it gives you a good feeling for how transcription works in terms of speed. That HHMI down there in the corner, that's Howard Hughes Medical Institute. And there are quite a number of individuals working, putting together these animations that you can find online. If you go and look for them, so you can just Google transcription and then ask for animations, and you can find lots of things to look at. So this is a rather quick one. Hopefully our sound is
Here the process begins.
This is sigma
Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene.
And you don't have to know that either.
Contact between the activator proteins and the initiation complex releases the copying mechanism.
So that's the
The
core
RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Again, it just gives you an idea of how fast this is happening. So what you see, that little ball, that's RNA polymerase, and it's just zipping along the DNA. What's coming in, the subunits, those are the RNTPs, and what's coming out the top is messenger RNA. And it's snaking off. However, I should specify all RNA is made the same way. Of course, we haven't talked about all the different types of RNA, but transcription is RNA synthesis, and all RNAs are made the same way. Now, an interesting twist on transcription, if you're talking prokaryotic cells, is that transcription is often polysystronic. And what that means is that multiple genes are copied at the same time. There will be one promoter site where a sigma factor binds. Again, the sigma factor is going to determine what part of the DNA is going to open up so transcription can occur. But in the prokaryotic cell, that promoter is in front of a whole series of genes, often 5 or 6 genes. So when transcription starts, you're going to make one long messenger RNA that isn't a copy of one gene, it's a copy of all the genes that are behind that promoter site. So you get one very long molecule. In eukaryotic cells, most of the time, transcription is monosystronic. And we're going to see why this is, because typically what happens is one messenger RNA is made, and then that messenger RNA gets modified. my understanding is that in eukaryotic cells, other types of RNA also get modified after they're built. So the transcription in eukaryotic cells tends to be one gene at a time, and then there's going to be some modification of the RNA that is produced. So polycistronic transcription is more efficient. It's going to allow these prokaryotic cells to either encode a whole series of enzymes or none of them. Typically what they're doing is transcribing a set of genes that will give them the enzymes to run a metabolic pathway. So we've seen the metabolic pathways: glycolysis, the Krebs cycle, the Calvin-Benson cycle. In order to run a metabolic pathway, you have to have all those enzymes. And so what prokaryotic cells will do is put all the genes, encoding all those enzymes, behind the same promoter. Sigma factor binds to that promoter. You're going to transcribe that whole set of genes. You're going to get a messenger RNA that can encode all of those enzymes. Either that or you get none of them. You either get to run the whole pathway or you get nothing. So it's extremely efficient. And again, as I said, in eukaryotic cells, sometimes genes are regulated together, but more often genes are regulated all by themselves. So they tend to be transcribed alone, and then their regulation is individual. Yeah, you get to learn about those exciting topics next time. Actually not. So this is showing you in a diagram the summary of replication. Again, what you need to know about replication: you need to know where does it happen. Replication happens wherever the DNA is, because replication is just copying DNA. You have to have enzymes that break the strands apart, break the hydrogen bonds, and then also prevent the DNA from getting tangled up. You have to have DNA polymerase III, the primary builder, but DNA polymerase cannot bind to single-stranded DNA. So to get DNA polymerase started, you have to have a primer, and primer is encoded. The primer, a short RNA segment, is encoded by the enzyme called primase. Once you have primers in place, then DNA polymerase can bind and begin to build. The leading strand is built as a continuous sequence. The lagging strand is built in a series of fragments, Okazaki fragments. And the Okazaki fragments have to be connected together, but first you have to take the RNA out. So DNA polymerase I does that, and then the fragments are connected together by ligase. Are all the enzymes in there? Actually not. All you've got is DNA polymerase and helicase. Oh well. This is talking about the PCR. PCR is what we're going to be doing in lab tonight, so rather than present this here, I'll talk about it in lab tonight. From here, we're going to go to the types of RNA, and our next process, which is translation. This little section has a quick review. Because we talked about DNA replication mostly last time, this is just a review of the whole thing. And it also has some images. So a quick review of information from last lecture, part of which was this lecture. What do we know about DNA? First of all, remember what the letters stand for. DNA, deoxyribonucleic acid. So it is a nucleic acid. It's a polymer made up of nucleotides. This other piece of kind of trivial information, this came out of a National Geographic magazine quite a number of years ago. It was determined that if you took all the DNA from all the cells in one human body, we have 46 chromosomes in most of our cells. If you take the DNA in the chromosomes and stretch it out, it would reach to the sun and back over 400 times. To the sun and back over 400 times. That is a lot of very long, skinny molecules. The information storage potential there is tremendous. And yet that DNA would fit in a box about the size of an ice cube. They didn't specify how big the ice cube was. It's like, well, not very big. I mean, we're not talking iceberg. We're talking ice cube. Molecules. So it's an amazing molecule, DNA. Again, tremendous information storage. This little film is going to take you through the process of DNA replication, again, as an animation. So it's going to show you all the different steps that I talked about last time, but this puts it together in an animation. Those things that look like, I don't know, maybe they look like Mike and Ike's except they're purple, those are supposed to be bacteria. So those are bacillus-shaped cells, and they're
Cells
undergoing
that
fission.
reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase 1 replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell.
So again, because they showed just one origin of replication, we have to assume that was a prokaryotic cell. Eukaryotic cells have multiple origins of replication. And from the point of origin, replication proceeds in both directions, away from the DNA. Away from the start point. You don't have one strand that is always the leading strand and one that is always lagging. They're opposite depending on which replication fork you're looking at. But that's kind of a nice summarizing movie. Now, DNA is DNA, although a little bit later on I'm going to talk about foreign DNA and ask you the question why. But anyway, for right now, DNA is DNA, but RNA is not all the same. So RNA, we said RNA molecules are smaller than DNA molecules. They have a different sugar, ribose instead of deoxyribose, and they have the base uracil instead of thymine. But RNA comes in different flavors, if you will. Actually, what it means is RNA molecules don't all have the same function. So, quick review about RNA again. RNA is ribonucleic acid. It is still a polymer, so it's a long chain of nucleotides connected together by phosphodiester bonds. It's just not nearly as long as DNA. The sugar is ribose. The bases are adenine, guanine, cytosine, and uracil. Uracil is a pyrimidine. The information that's carried in RNA molecules at this point in time, most of it, if we're talking cellular organisms, is encoded by DNA. But biologists are pretty certain that RNA evolved first, and that the original information carrying nucleic acid was RNA, not DNA. But anyway, so RNA's structure is determined by the nucleotide sequence of DNA, unless you're talking viruses, because some RNA in viruses is encoded by other RNA, and some RNA in viruses is encoded by, some DNA in viruses is encoded by RNA. The whole thing gets very complex. So, the process of making RNA using DNA as a template, RNA synthesis, is called what? Transcription. We're just copying information. Transcription is just rewriting the information. You're not changing the language. It's still nucleic acid. Copying one nucleic acid, deoxyribonucleic acid, into the other ribonucleic acid. Into the other, which is ribonucleic acid. So again, that's transcription. It requires DNA as a template. It requires enzymes. It requires energy. We already watched that. We're not going to watch it again. The subunits that were coming in at the bottom as that DNA polymerase was zipping along, those subunits were the nucleoside triphosphates. Are they DNTPs or RNTPs? RNTPs. So the nucleoside triphosphates, if you're doing transcription, are RNTPs. The sugar is ribose. And you have adenine, guanine, cytosine, and uracil, not thymine. Transcription factors, again, the most important one is sigma factor. Sigma factor has to determine where transcription is going to start. Sigma factor binds to a promoter site. And then, as I said, in prokaryotic cells, transcription is mostly polycistronic, whereas in eukaryotic cells, it's mostly monocistronic. Sigma factor is actually part of RNA polymerase. Remember that conglomeration that I showed you in the first place, where the sigma factor was the pink part? So you have alpha, alpha, beta prime, beta prime, and omega, and sigma. So it's part of RNA polymerase, but it's not part of the core enzyme. It just gets the process started, then it goes away. So RNA molecules are all copies of information stored in DNA, but the information varies, and what RNA molecules do varies. So we're going to learn about some different types of RNA. What's interesting here is that the information has been changing over time. Used to be there were just 3 types of RNA. And you could say prokaryotic cells, eukaryotic cells, it doesn't matter, there's just 3 types. That's not the case anymore. Eukaryotic cells have at least 5 different types. And prokaryotic cells, we don't really know, because now the whole CRISPR business has come online and it's very complex. Typically, RNA molecules are designated different varieties by a single letter in front. There used to be a single small letter and a dash in front of the RNA. They've taken out the dash now, so messenger RNA is just small m RNA. Messenger RNA gets its name because it carries the message telling the cell what protein to make. It's a copy of segments of DNA that are called structural genes. The DNA is in the nucleus, or in the nucleoid, or associated with plasmids, etc. But that information is kind of protected. Meanwhile, ribosomes are some distance away, and that's where protein synthesis is going to take place. So the messenger RNA is taking the message, the nucleotide sequence, from where the DNA is, out to the construction site, out to the ribosome, telling the cell which protein they get to make. Messenger RNA molecules have a relatively short life expectancy, supposedly in eukaryotic cells about 2 minutes. Why? Because there are enzymes called RNases, and RNases degrade RNA very quickly. If you work in a research laboratory where you're having to work with RNA, apparently it's really difficult because there's RNase enzymes all over the place, and your RNA can very quickly be degraded. So messenger RNA has a short life expectancy, so it's constantly being made. The cell is making RNA polymerase. That's one of your constitutive enzymes, because they're always making RNA. To give you an idea of how long RNA is, RNA, like I said, is much shorter than DNA. Typically, proteins in eukaryotic cells, about 361 amino acids. In bacteria, about 267. In archaea, 247. I don't know who does this calculation. It's like, really? And we don't really know this because we only know about somewhere between 1 and 10% of the prokaryotes at this point. So we can kind of say this, but is it real? The significance of this is that there's 3 nucleotides encoding every amino acid that's in a protein. So if a protein is 361 amino acids, the RNA that encodes that protein is 3 times that long at least. So it's going to be longer. So it tells you again, even though messenger RNA is not considered huge, it's a pretty long chain of nucleotides. So they have to be longer than the proteins that they encode. So messenger RNA carries the message telling the cell what protein to make. Ribosomal RNA is associated with ribosomes. We spend a lot of time talking about prokaryotic ribosomal RNA because when you do the physiological unknown 2, you're going to be identifying your bacteria based on the 16S ribosomal RNA nucleotide sequences. The reason we use nucleotide sequences of ribosomal RNA is because all cells have to have ribosomes. They don't make proteins without ribosomes. If you mess around with the sequence of RNA that builds the ribosome, then protein synthesis isn't going to happen. Consequently, that type of RNA is very highly conserved over time. It hasn't changed much over evolutionary time. It has changed some, and the changes allow us to distinguish one type of bacteria from another. Ribosomal RNA in prokaryotic cells comes in 3 lengths. So there are 23S, 16S, and 5S. Remember, the S is a Svedberg unit, and it's a measure of density. The 16S is actually between 1,500 and 1,600 bases long. The longest ones that we've ever gotten are somewhere around 1,533 bases, so 1,533 bases long. So they're pretty long. The 5S are smaller, the 23S are larger. Now we know that ribosomes only come together, the 2 subunits of the ribosome only come together during protein synthesis. So when you look at the ribosome, the small ribosomal subunit, which in prokaryotic cells is the 30S subunit, that's where we find the 16S ribosomal RNA. And that 16S ribosomal RNA is associated with proteins. There's about 21 different proteins, so you have a whole conglomeration. These diagrams show the small subunit is just this purple blob. but it's really very complex. So the RNA is wound around these proteins in a very specific way. The larger subunit is made up of the two other RNA types, so the 23S and the 5S are jointly there. Another interesting thing about ribosomal RNA, there is an operon, which is a unit that controls the synthesis of ribosomal RNA, and all the genes encoding the ribosomal RNAs are on the same operon, so they're all regulated together. You've got the 16S, the 23S, the 5S in a series. So if you make any of them, you make all of them. Again, transcription is polycistronic. That complete ribosome, 30S unit, 50S unit, come together only when protein synthesis is happening. So ribosomal RNA joins with proteins to make the ribosomes. And the 23S is a ribozyme. That ribozyme is called peptidyltransferase. For a long time, protein synthesis was credited to an enzyme. When I say protein synthesis, I mean the catalytic reaction binding amino acids together. Researchers knew that something was catalyzing the formation of peptide bonds. They assumed it was an enzyme, and they named it peptidyltransferase. So it has the same enzyme ending, that ASA ending, but it turns out that peptidyltransferase isn't an enzyme at all, it's a ribozyme. It's the 23S ribosomal RNA. This is just showing you how the RNA is tangled up with the proteins within the ribosomal subunit. It's quite complex. So again, that ribozyme, the 23S ribosomal RNA, is going to catalyze the formation of peptide bonds between amino acids. You'll see in a minute why it's called peptidyltransferase. It's actually transferring a peptide bond from one transfer RNA to another, which maybe doesn't make any sense right now, but it will. Okay, so messenger RNA carries the message, ribosomal RNA builds the ribosome with proteins. The third type of RNA is called transfer RNA, small t RNA. These are relatively little as RNA molecules go, so they're only about 74 to 93 nucleotides long, and their job is to carry individual amino acids to the ribosome for protein synthesis. Typically, transfer RNA molecules are illustrated looking like a clover leaf, where this is the stem of the clover leaf, and you have a leaf going this way and a leaf down here and a leaf over here. Well, all of these things have different names to them, which for you guys, you don't need to worry too much about, except you need to know where the anticodon region is. So notice that the other thing about transfer RNA, it folds back on itself, and where it folds back, it hydrogen bonds. Complementary sequences hydrogen bond. So there are complementary sequences here in the stem, and here, and here, and here. And they're actually twisted like DNA, so they form a helix, they're not straight. The other thing about transfer RNA is that the top of the stem, typically there's a sequence that reads ACC from the 3' end. That's where the amino acid is going to attach. So our transfer RNA, this kind of weird clover-like shape, up here at the top of the stem is where the amino acid is going to attach. And then we have loops off to the side that are referred to as T loops and D loops. Don't spend a lot of time deciding which side is T and which side is D, because it depends on where you are relative to the transfer RNA. So if you're looking at this diagram from this side, you could say, okay, the D is on the left. But if you were looking at it from the other side, the D is on the right. So never mind. You need to know that the top is where the amino acid binds, and you need to know about this little sequence down here. This is called the anticodon region. The anticodon region plays a very significant role because it's going to determine which transfer RNA can bind to the messenger RNA at the ribosome. Again, the ribosome is the site of what? Protein synthesis. What determines the sequence of amino acids in a protein is the sequence of nucleotides in messenger RNA. Messenger RNA has to go to the ribosome. The ribosome is made of ribosomal RNA plus protein. Transfer RNAs bring the amino acids down. So these three types of RNA are all playing a role in protein synthesis. So here's an interesting question, and you should be able to answer this, except that it requires that you think about something that was on last time's quiz. So how does a cell combine a specific transfer RNA molecule to a specific amino acid? But you can think about it this way. How does a cell catalyze a synthesis reaction where you're binding this unit and this unit, putting them together? You gotta have a catalyst, right? Your catalyst can either be a ribozyme or it's going to be an enzyme. What I can tell you is this specific reaction, binding an amino acid to transfer RNA, involves an enzyme. What's the enzyme called? It's aminoacyl tRNA synthetase, because it's synthesis. It's binding an amino acid to a transfer RNA. So there it is, that doggone enzyme. Last time I gave you the name, it says, what does it do? Now I'm asking you to think about, it's an enzyme. What's the name of it? It's binding an amino acid to a transfer RNA. So this particular enzyme has to be able to recognize the amino acid it's going to bind. It has to be able to recognize a specific transfer RNA. And it also has to have a site to bind an energy source. It binds to ATP because connecting an amino acid to a transfer RNA requires energy. It's a synthesis reaction. ATP provides the energy, and those two are connected together. So again, aminoacyl tRNA synthetase. That's the enzyme that binds an amino acid to a transfer RNA. And in this diagrammatic representation, it just shows you the transfer RNA binding at one site, the amino acid binding to another site, and then that little site in between is where the ATP binds to provide the energy to connect them together. This thing that comes off, technically this is called aminoacyl tRNA. Here's the tRNA, here's the amino acid. Sometimes references want to call these charged tRNAs. To me that's like, why? Just call it what it is. It's an aminoacyl tRNA. So the transfer RNA with its amino acid on it. This is the anticodon region. The function of this transfer RNA is going to be to bring that specific amino acid to the ribosome, and it's got to put it in the right place. And the way it knows where to bind is it's going to attach to the messenger RNA. This little region here is called a codon, and the codon has to be complementary to the anticodon. These two are going to attach to one another. What kind of bonds? Hydrogen bonds. We're talking about pairing bases. Anytime you put bases together, you're going to form hydrogen bonds. Now, this is a kind of a recent one. So prokaryotic cells have messenger RNA, they have ribosomal RNA, they have transfer RNA. And for a long time, it was thought that's all prokaryotic cells have. Fairly recently, there's another type of RNA found in prokaryotic cells, and it's called cRNA. Small r, small c, small r, RNA. The CR stands for CRISPR. For the prokaryotic cell, this is an immune system, if you will, because what the cell is doing is taking segments of DNA from viruses. They're stealing pieces of DNA from viruses, and they're bringing them in and actually attaching them to their genomes. Researchers are finding out that archaea apparently have very long collections of viral DNA that they're adding to their chromosome. Bacteria have a few less, and these things are not static, so they'll bring these viral chunks in and they keep them for a while. They can discard them and get new ones, so it's kind of an exchange going on. But those pieces of viral DNA encode the cRNA, the CRISPR RNA. So then researchers wanted to know why. What are they doing with these things? These little RNAs, these little CRISPR RNAs, can bind with, hydrogen bond with, specific viral DNA if it gets into the cell. And what these little CRISPR RNAs do is they take a type of protein that the cell makes, which is called a Cas protein, which means CRISPR-associated protein, CAS. Anyway, the Cas proteins will be taken to the viral DNA, should the virus get in again, and it'll take this Cas protein there, and the Cas protein cuts. So it's how the cell defends itself against viruses. We're going to find out later on it's not the only mechanism they have, but this one is super sophisticated, where they're actually taking pieces of the virus and then using the viral genes to encode their own RNA, these little short CRISPR RNAs are guide RNAs. And what they do is guide cellular proteins to the viral RNA, or excuse me, viral DNA if it gets in again, and then they chop it up.
I think HiGen's locked out.
Huh?
I think HiGen is locked out.
Oh. Thank you.
You're welcome.
Sorry.
Sorry.
So, the reason that door is locked periodically is because the instructor that's in this building before me has listened to the emergency drill situation, so that if there's an emergency lockdown, the instructors are supposed to make sure that the door is locked. Well, you can't lock that door from the inside. She doesn't want to go out, so she just locks it. Then, you know, then you have to pay attention to who goes out because they can't get back in. Now it's unlocked. Should there be a shooter in the hallway, somebody's going to have to go out there and lock that door. It have to be me, because nobody else can be sent out there. So while you guys are hiding in here, I'll sneak out there.
We'll just take them out.
Yeah.
We'll pour some bacteria down.
Well, anyway, what's really fun about this CRISPR thing, there was a big court case about it. Who discovered the CRISPR? Who got to use the CRISPR? Because scientists can synthesize nucleic acid. I'm not sure exactly how they do it, but they can. If you send a nucleotide sequence to a company that makes nucleic acid, they will just use whatever nucleotide sequence that you send them, and they'll synthesize that. So companies can synthesize CRISPR RNAs that can target any DNA that's in a cell. Now, if you're talking not prokaryotic cells, but eukaryotic cells, this gives scientists a very specific tool for cutting DNA in a very specific place. So they can use these little CRISPR RNAs, and then the CRISPR RNAs will take the Cas proteins, and the Cas proteins will cut the DNA. It's given scientists a tool for genetic manipulation. And this is just starting to be used, but it's a big, big deal. So you probably hear more about CRISPR and Cas9. Cas9 is the protein that cuts. Well, I'm thinking about it. The CRISPR-associated proteins, there's a bunch of them. Cas9 is just one that they found that is particularly useful. So it's in the news. Anyway, so we're all interested in the CRISPR-Cas9 system. Now, eukaryotic cells have other types of RNA. They have messenger, they have ribosomal, they have transfer, and then they have these other RNA types that apparently are not in prokaryotic cells. Well, prokaryotics have CRISPR, that apparently eukaryotics don't. One of the types that eukaryotic cells have is called small or short RNA. And the letter that goes in front, in this case, is an S, which is kind of interesting because initially transfer RNA, because it was little, they wanted to call that one soluble. And it's a good thing they didn't, because then they'd have too many S's. So transfer is T, S is short. Short or small. These are the RNAs that are associated with spliceosomes. Remember we talking about spliceosomes? We're talking about eukaryotic cells. These are basically little globules of small RNA and protein. They're in the nucleus, and their job is post-transcriptional modification. So in eukaryotic cells, after transcription happens, post-transcriptional, when transcription happens, you have an RNA transcript. That RNA transcript is going to be modified, and it's modified by spliceosomes. And what the spliceosomes do is they take out sections of RNA that are called introns. Intron stands for intervening regions. Turns out in eukaryotic cells, sometimes more than half of a messenger RNA transcript is intron. Which kind of brings up the question, why? Why are eukaryotic cells so inefficient? Why do they have so much RNA? I mean, they have to make it, and then these pieces get cut out. What scientists are asking now is, where do they go? What are they used for? We don't necessarily know. But the introns get removed, and then sections that are called exons, notice the spelling, there's not two X's, there's only one. Exon stands for expressed region. The exons get connected together. How many of you know what splicing is? splicing means putting things together. It's not a term that we use a lot in our language anymore, but people used to splice ropes. They would actually weave the strands of the ropes and splice ropes together. Splicing is sticking things together. So a splice of sewn is splicing together exons. You have to take the introns out first, and then they connect all these exons together, the expressed regions. They're going to end up with a much shorter messenger RNA, but that's the RNA that's going to code for a specific protein. So these expressed regions. That's not the only thing that happens during post-transcriptional modification. There's also a cap that's added. Typically it's a modified guanine molecule, methylated guanine, and then there's a tail of polyadenine, a whole string of adenine nucleotides, which kind of makes sense if you think about it because ATP is the most common high-energy compound. So they make this big long adenine tail. This is supposedly going to help to stabilize the RNA. Again, there's lots of RNase enzymes. They're taking RNA apart. Messenger RNAs don't last very long, but if they have this tail, they last a little bit longer. So it's going to help them stay around a little bit longer. And then there are RNAs called microRNAs. MicroRNAs, miRNA. So these guys are really little, only about 20 bases in length, slightly more than that. Very tiny little things. But apparently these can regulate gene expression. For our purposes, that's all you need to know. Exactly how do they regulate gene expression? Don't worry about it. But this is a big deal, because if you think about it, we all start from one egg, a zygote, a fertilized egg cell. That cell undergoes cell division, but you don't end up with just a mass of the same cell. Instead, we have differentiation. So you end up with some muscle cells and some nerve cells and some blood cells, etc., etc. That means some of the DNA gets turned off and some of the DNA is allowed to be used. For a long time, researchers going, how does some of the DNA get turned off? Apparently the answer is microRNA. So the microRNAs are involved in regulating which genes get expressed and which ones don't. So this is a big deal. If the microRNAs are bound to portions of RNA, then they're not going to be translated. They're not going to be used to make protein. So these things are kind of floating around, regulating which genes get expressed. When we talk about gene expression, what we mean is genes being copied into messenger RNA, and then the messenger RNA being used to make a protein. So the microRNAs prevent that. Or sometimes they cut the messenger RNA, or they can take the tails off of them, and then they're just destabilized, and the RNase enzymes chop them up. Or they can just interfere with translation, which we'll get to in a minute. Well, so then researchers have constructed yet another type of RNA. As far as I know, the siRNA is synthetic. Again, these are really little, they're tiny little RNA molecules that can be used to purposefully silence genes. So they're tools that people can use to do genetic manipulation. So they're going to block gene expression. Okay, now we're moving to our next topic, translation. And once again, I'll ask you, what does that word mean? If you're not in a biology class, what does it mean to translate something?
Take information and make it something else.
Typically, translation, if you're looking at information, but typically you're changing language, right? When you translate, you're reading something that's in one language and you are deciphering it and rewriting it in a different language. This is important because transcription, you didn't change language. Transcription, it's nucleic acid and DNA copied into nucleic acid that is RNA. Now we're going to translate. We're going to change languages. Again, don't mix these terms up. And I can tell you this, and students do it, and it's like, no, you know what translation means. Changing languages. Translation then is copying information, rewriting it in another language. You're still going to have the original meaning, but you're going to have a different language used. In a biological sense, what we're translating is the information stored in RNA molecules, specifically messenger RNA. That messenger RNA nucleotide sequence is going to be copied into an amino acid sequence. We're changing language from nucleic acid to protein. Remember where I said it's very important that you understand what proteins are made of? Proteins are made of amino acids. Nucleic acids are made of nucleotides. So when you change language, you're taking a nucleotide sequence that is messenger RNA and you're converting it, actually you're not converting it, you're using it to build an amino acid sequence, a protein. That's another mistake students tend to do. They'll write DNA gets converted to RNA. No, it doesn't. The information in DNA is copied to make RNA. And in translation, the information stored in messenger RNA is copied to make an amino acid sequence, a protein. So we're changing languages. Where does this process happen? Translation happens. Oh, you gotta be more specific than that.
Ribosome.
At the ribosomes. Ribosomes are the site. Protein synthesis. Translation is protein synthesis. Copying information that's stored in messenger RNA into an amino acid sequence. What's going to happen at the ribosome? I know this is tough because a lot of you are trying to study for the lab exam. Just for right now, stop it. This topic really bites people, and it shouldn't, because it's pretty straightforward. But unfortunately, a lot of people go back and they look at their notes and it's just like, well, I don't know what any of this means. You need to know, okay? So translation occurs at the ribosome, and the reason it occurs at the ribosome is because that's where peptidotransferase is. Peptidotransferase is what catalyzes the formation of the peptide bonds between the amino acids, and peptidotransferase is ribozyme, 23S ribosomal RNA, in prokaryotes. In eukaryotes, it's a different number. So proteins, or in some cases just polypeptides, because sometimes a protein is only one polypeptide, and sometimes a protein is multiple polypeptides. But anyway, proteins, polypeptides, they're made up of amino acids, and there's only about 20 different kinds that are used to make proteins. That's another good thing about this class. It's not a biochemistry class. You don't have to memorize all the amino acids. But you might recognize a few of them. Well, so for a long time, chemists thought nucleic acids could not possibly be the information within cells because they were just too simple. All you had was a sugar, a phosphate, and 4 nitrogenous bases. And they knew that proteins had these big, long strings of amino acids. If you've got 20 different amino acids in a protein, there's no way that each base can code for one amino acid. There's not enough. So they knew right away that individual bases, adenine, guanine, cytosine, uracil, could not code for a single amino acid. They thought, well, maybe if you take the bases 2 at a time, so read them in pairs. They found out if you put them together in all the possible combinations, you come up with only 16 possible. Still not enough. If you've got 20 different amino acids, 16 possible combinations, not enough. So they said, how about if the nucleotide sequence is read in sets of 3? If you take the nucleotides in sets of 3, or bases in sets of 3, you have 64 possible combinations. Sounds kind of far-fetched, but it turned out that's how the sequence is read. So when a cell is determining what nucleotide sequence encodes what amino acid sequence, the cell has to take the nucleotide sequence in sets of 3 bases. A set of 3 bases is called a codon, and there are 64 possible. You see them showing up here. This thing is sometimes referred to as the genetic code. And as soon as I show you that, you can find lots of different versions of this online, lots of different representations. And what I can tell you is, although most cells use something that's very similar, there are some exceptions. So there are some variations. And apparently mitochondria have some really weird changes in their genetic code, and never mind. Here's another way to express it. So here's the genetic code again, showing bases in sets of 3. In this one, you have the first base in the middle, and then the second base, and then the third base. So there's always 3 bases, and 3 bases encode a specific amino acid. In this case, glycine, alanine, etc. Around that ring. Again, there's lots of different ways to represent this. How did they figure this out? Basically what they did was take bases in sets of 3 where they had all the same bases. So for instance, you could take AAA, actually, I don't know what AAA encodes, UUU. You take polyuracil, and they made up a whole chain of just polyuracil, and then ran it through a cell to see what it would encode, and it was phenylalanine, just a whole chain of phenylalanine, one after another. So they found out that UUU encodes phenylalanine. Anyway, these sets of 3 bases are called codons, and this is significant. A set of 3 bases on messenger RNA is a codon. A set of 3 bases on DNA is a codon. A set of 3 bases on transfer RNA is an anticodon, and those are going to come together when transfer RNA binds with messenger RNA at the ribosome. Do you need to memorize the genetic code? No. And don't listen to anybody that tells you that you do. But you do need to know how to use the genetic code. There's another homework assignment. So I did mention to you guys there's a homework assignment about glycolysis and cellular respiration. There's another homework assignment coming up about DNA, RNA transcription, translation, and it requires that you translate on paper. Translation is protein synthesis. So you have to be able to read the genetic code. It turns out that with 64 codons, there's going to be a lot of overlap. So many codons will encode the same amino acid. We sometimes say that the genetic code is redundant. There is overlap. Most often there's 4 to 6 different codons that will code the same amino acid. But there's also a specific codon that is the start. It's called an initiator codon, which is AUG. So on messenger RNA, an AUG is a start codon. AUG also encodes the amino acid methionine. So if it's at the start, it encodes methionine, but if it's out in the middle someplace, it still encodes methionine. And then there are 3 codons that for a long time people referred to them as nonsense codons. And those are the codons UAA, UAG, and UGA. And the reason they were called nonsense is because what researchers found out is they didn't encode anything. That is, no amino acid. So they called them nonsense, and then after a while they figured out they're not nonsense, they're stopped. They're like a 0. They have to be there. They tell the cell to stop translation. That's where the building process is going to stop. Those 3-letter sequences, UAA, UAG, UGA, all have to do with somebody going away or being gone. So UGA is you go away, UAG is you are gone, and UAA is you are away. So you can kind of remember them that way, in case you need to remember them. They also have color names associated with them. They were apparently initially referred to as ochre, amber, and umber. My understanding is amber was somebody's significant other, and then they just made the other 2 colored names to go along with it. You don't see those names used very often anymore. So terminator codons, they signal stop. Now translation is broken into a series of steps or stages. This is true for replication and transcription. I don't typically test on what are the stages. You need to know what the process is in general. But in this case, the initiation of translation, because translation requires the ribosome, the first part is binding one ribosomal subunit to messenger RNA. It's the small ribosomal subunit, the 30S in prokaryotic cells, the 40S in eukaryotic cells. It has to bind to messenger RNA, and it's going to bind somewhere upstream of the start codon. Sometimes this involves a sequence called the Shine-Delgarno sequence. Never mind, you guys don't have to know that kind of detail. Again, the sequence where this thing binds is fairly lengthy. It's not just 1 or 2 bases. But the next thing that's going to happen, well, this is showing how does it bind. It's actually the 16S ribosomal RNA, which is part of that small subunit. That 16S nucleotides or bases on the 16S have to bind with specific nucleotides on the messenger RNA to hold that thing in place. So again, it's complementary base pairing, and here you see this base pairing. This is the messenger RNA down here, and it's actually binding to a nucleotide sequence on that 16S. So it's going to hold it in place. Again, That's more detail than you need to know. Don't worry about Scheindel-Garno sequences. But what kind of bonds? Hydrogen. Anytime you have bases coming together, that's hydrogen bonding. A with T or A with U, C with G. Once the small ribosomal subunit is bound, then the second step is going to be binding a specific transfer RNA that's carrying the amino acid methionine, because the ribosomal subunit is going to move till it gets to the start codon, the AUG. AUG encodes methionine, but methionine doesn't come down by itself. Methionine is carried down by a transfer RNA. So that transfer RNA has to have a sequence that's complementary to the codon at that specific location. That methionine, it's methionine in eukaryotic cells, it's going to be bound. In prokaryotic cells, it's N-formal methionine, not to worry about. So AUG is a start codon, just know that it encodes methionine. Once you get the transfer RNA holding methionine on the start codon, the AUG, then the second ribosomal subunit can bind. And then it's going to start the translation process. So it requires putting together the three types of RNA: the messenger RNA, which carries the message, the ribosomal RNA that is part of the ribosome, and the transfer RNA that's carrying the amino acid. You've got to get them all in there to get this process started. This is showing the initiation. Here's the small ribosomal subunit. This is the messenger RNA. The small ribosomal subunit has the 16S. And then this is the transfer RNA carrying the methionine. And then the second ribosomal subunit binds, and the methionine is sticking up there. The other thing that happens, if you look at the ribosome, typically there's 3 different positions on that ribosome where things can bind. They're referred to as the A site, the P site, and the E site. The A site is the amino acid binding site, so this is where new transfer RNAs are going to come in carrying their amino acid. The center spot is called the P site, which stands for polypeptide. Right now it's just one amino acid, so it's not a polypeptide. And the E site is exit. So as the ribosome moves along this direction, these sites are constantly moving along with it, and you're reading this sequence on the messenger RNA as transfer RNAs come in. Each transfer RNA comes in to an A site. So translation involves, first step, binding the small ribosomal subunit to the messenger. Second step, binding the transfer RNA carrying methionine. The third step, binding the second half of the ribosome. And then we're ready to start translation. Elongation is then just adding more adding more amino acids, bringing them in one at a time. Each amino acid is carried by a transfer RNA. They come into the A site, and what's in the P site is the polypeptide. So again, this is representing transfer RNA. This first one is carrying methionine. The next one that can come in is determined by this sequence on the messenger RNA. Whatever this nucleotide sequence is, the anticodon has to be complementary. And that specific amino acid transfer RNA that comes in there is going to be carrying a specific amino acid. None of it is random. And then the E site is where they go out. So again, A is amino acid binding site, P is polypeptide binding site, E is exit site. When the transfer RNAs come into the amino acid binding site, the anticodon on the transfer RNA has to bind with the codon of the messenger RNA, and that's going to be hydrogen bonding. So hydrogen bonding between transfer RNA and messenger RNA makes sure that the right amino acid is in the right spot, in a sense. But what determined that the transfer RNA was carrying the right amino acid in the first place was peptidotransferase, which is an enzyme. No, it's aminoacyl tRNA synthetase, which is an enzyme. Peptidotransferase is a ribosome. Notice all these diagrams keep changing. There are many, many different images that you can find if you go looking for this process online. So here you see it again. This is the ribosomal small subunit. This is the ribosomal larger subunit. Here the transfer RNAs are these weird looking things. They're going to come in. This is the anticodon region. The anti-codon region has to be complementary to a codon, set of 3 bases on the messenger RNA. That anti-codon-codon hydrogen bonding makes sure that the transfer gets in the right place, which is going to make sure that the amino acid sequence is correct. Now, why is the ribozyme called peptidyltransferase? Because what it's going to be doing is binding this peptide chain to this amino acid. What it does is transfer the amino acid, transfer the peptide chain from this transfer RNA over to here, and then this whole thing moves over. So this is going to be in the P site. It's holding the polypeptide. But it's called peptidyltransferase because it's transferring the little peptide from one transfer RNA to the next. And again, peptidyltransferase is a ribozyme. So this is again just showing you animations. The peptide chain is growing. We're adding more amino acids up here, growing the peptide. New ones are constantly coming into the A site. The other thing about this is once that transfer occurs, so that the polypeptide is transferred to a new transfer RNA, the transfer RNA that is released can go back and pick up another amino acid. They can be used over and over again. I don't know how long they last, but they're not immediately degraded. Again, lots of different versions showing this process. These things are coming in. They come in, transfer RNA now looking like a stick. Each transfer RNA brings in one amino acid, and then this chain gets transferred onto that one. That's what the enzyme does. It's actually building a bond between those 2 amino acids, but it takes it off of this transfer RNA, which then exits, transfers it to this one, and this one moves into the P site, which is where the polypeptide is. Another version of it, showing the same thing, showing transfer RNA going off on the left, transfer RNA coming in on the right, bringing a specific amino acid with it. The last part, the termination, is going to happen when you get to a terminator codon, the stop codons, because the stop codons don't encode any amino acid. When the cell gets to that point, then the peptide chain that's been being built is going to come off, and the process is finished. Then the ribosomal subunits come apart. That's the end. Again, these messenger RNA molecules have a relatively short lifespan. Cells have quite a large number of ribosomes, so oftentimes what they will do is attach more than one ribosome to the same messenger RNA so that they're all building at the same time. Not quite in the same place, but they're just going to move along the messenger RNA. This diagrammatic representation shows what's going on here. So we have a whole string of ribosomes attached to the same messenger RNA. This one has been translating the longest. This one's finished. Here's its polypeptide being released. These are all moving along in this direction. This one's just getting started, so it's attaching. It hasn't built anything yet. That chain of ribosomes attached to and translating the same messenger RNA has a name. If you have many ribosomes, what word might you use? What's our word for many? Poly. So a polyribosome. Sometimes it's called a polyribosome, sometimes it's just called a polysome. So if you read about a polysome, what is it? It's many ribosomes. They're attached to the same messenger RNA, translating it. And they're all ultimately going to produce the same polypeptide. Chain's going to be the same. This is showing you an electron photomicrograph. So these have actually been visualized. Here's the messenger RNA, this little light string. And these are polypeptide chains being built associated with ribosomes that are moving along with me. And then, as I said, in prokaryotic cells, you have polyribosomes, but you also have polycistronic transcription. Don't mix those two words up, polyribosome and polycistronic. Polycistronic transcription means you're going to encode multiple different proteins on the same section of messenger RNA. So it's got more than, it's a copy of more than one gene. Now this is going to show the translation process in an animation.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA.
Actually, it's determined by
Complementary
the enzyme.
base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified,
which is post-translational modification. We're not talking about that. There was one statement that the narrator made, and that was that the anticodon determines which amino acid the transfer RNA is carrying. That's not true. What determines which amino acid goes on which transfer RNA is the enzyme, amino acyl tRNA synthetase, and as far as I know, it's not looking at the anticodon. It's looking at some other part of that transfer RNA molecule. It is significant, though, when you start doing the homework assignment, when you look at the way the translation process works, you have the messenger RNA codons, and you have transfer RNA anticodons. All of those are letters, nucleotide bases. When you translate on paper, you cannot incorporate the anticodons. It's just going to mess you up. You have to look at the genetic code, and you have to say, this codon encodes that amino acid, and leave the anticodons out. Don't bring those into play, because it just messes you up. So you're supposed to practice this translation process on paper. It's a homework assignment. Again, it's DNA, RNA, protein synthesis. It has a little bit of information about operons, which we haven't gotten to yet. But ignore the anticodons when you translate on paper. You're just reading the messenger RNA nucleotide sequence, set of 3 bases at a time, and each codon codes for a specific amino acid according to the genetic code. Ignore the anticodons. They have to be there for the cell, but not for you guys. Oh gosh, I forgot to look at the clock. Oh, that's okay. We're done.
Longer break.
The good news is, yeah, you can take a longer break if you want, because we don't have a quiz tonight. I have to go get it. I'm just gonna give it to you, and you're gonna take it away and do it on your leisure time, not between now and Thursday, because between now and Thursday, you have to study. What did you use the following Tuesday? Sure. Yeah, don't hang on to it for too long, because, you know, then we'll have another one. They just keep piling up. So go ahead and take a break. We'll start lab maybe a quarter after. So I have to go get